So let me start with a few announcements. First one is lab this week. Actually, through the rest of the term, lab this is going to start being even more informal than usual. So we're at the point where you just have to come in and count flies, count lots and lots of flies. Some of you aren't quite there yet. Just keep an eye on your fly bottles. When you start getting adults, collect them and count them. In the last experiment, I sort of gave you a limit. I said, okay, if you can get a couple hundred flies, you're good. That's fine. For this experiment, for the mapping cross, we need lots of flies because we're looking for some rare events. So that means I'm just going to tell you, okay, look at the calendar. Once you start getting adults from the F2s, the F2 adults, you're going to have about 10 days to count. Because after 10 days, you start seeing a third generation emerging. So just keep counting until then. So yeah, if you can count 10,000 flies in your group alone, that would be terrific. I don't expect that, but just count lots of flies. I mentioned a little write-up on the Canvas page that there are ways you can do this. When I say, you get adults start counting for 10 days, that doesn't mean come in every day and count your little brains out trying to keep track of all these flies. You could just say, okay, I'm going to come in periodically every couple of days, in a week, whatever. And then you'll just have a whole pile of flies that you have to count all at once. It's a massive effort, but however you want to work it, that's fine. Okay, then let's see, next announcement. The exam. I am working on the exam. I was very generous in giving extensions to a lot of people, so I can't get it back to you today. But Wednesday, I know there were some questions about some of the problems because I made them a little trickier than usual this time. So we'll go over those on Wednesday. I should be able to free up some time Wednesday for that. All right, so any questions? We're starting chapter 9 today. Extrachromosomal Inheritance, there it is right there. After that, we're going to take a big leap forward. We're going to go to chapter 23 and talk about developmental genetics for a while, because that's the stuff I really, really like. Yeah, this class, it's all about me, so that's what we're going to do. Okay, so extrachromosomal inheritance, what does that mean? Well, we've been talking about nuclear genes so far, right? You remember from cell biology, you got a nucleus with all those chromosomes in it. We've been talking about genes on chromosomes in the nucleus. Those often fit, but not always, Mendelian inheritance rules. But you know from cell biology again that there are these other organelles in the cell, in particular mitochondria and chloroplasts, that also contribute to the phenotype of the organism. And those are inherited with slightly different rules. So we're going to talk about organelle and heredity. The other thing to consider is that what gets passed on to the offspring isn't just a nucleus and a mitochondrion, right? There's a whole bunch of cytoplasmic factors that get passed along too. And those can contribute in interesting ways to the phenotype of the progeny. So that's where maternal effect and imprinting come into play. And then there's also infectious heredity. Turns out that there are heritable diseases, viruses and bacteria, that can inhabit an egg and get passed on to the offspring. So lots of different ways besides just passing on chromosomes from the nucleus to generate new phenotypes. All right, there's a couple of properties to know about these. So one is reciprocal crosses matter. This is largely because in animals at least, typically the female produces a large gamete. It's got a large cytoplasm, got lots of gooey stuff in there. And the male produces stripped down gametes that contribute next to nothing. So we see a difference in the sexes here. Another common characteristic is that the project show the characteristics of the maternal parent only. So as you probably remember from cell biology, your mitochondria came from your mother, but not from your dad. So that makes a difference. Another thing, genes can't be mapped. So here we are in the middle of this nice mapping experiment, right? That's what you're doing in the lab, is you're mapping out the locations of white, miniature, and forked. If we throw in some of these mitochondrial or chloroplast genes, you can't do it. They're not linked. There's no way to measure a link between nuclear genes and these other genes. And then the other thing is, when you do these crosses, no, Mendel goes right out the window, sorry. No 3-to-1 ratios. You don't have dominance. You have some other complicated phenomena we'll get to in a moment. And you don't have independent assortment. It's just a different set of rules behind these patterns of inheritance. Okay, probably the best one among us humans is this, mitochondrial inheritance. But if you were a plant, you'd also care about chloroplast inheritance. We don't have chloroplast, so forget that for us, but for lots of organisms, it's very important. A couple of key things about these organelles, as each one has their own circular strand of DNA. Again, you remember that from cell biology. They're basically prokaryotes that got incorporated into the eukaryotic cell and have their own genetic contributions to make. Both of these organelles transcribe RNA. They have independent protein synthesis mechanisms. They have their own ribosomes. So they're biochemically different in that sense from the rest of the cell. They're independently replicating. They divide to form new organelles independently of cell division. Yeah, so you got this big cell, right? And it's going to go through cell division. We can see that in the microscope. We watch it go through mitosis or meiosis or whatever. There are also mitochondria or chloroplasts in there that are dividing on their own clock. They will make new mitochondria in there, and that's independent of the cell cycle pattern. Now, despite having their own DNA, both also rely on nuclear genes, and we'll get into that in a little bit. So, you've got a set of genes on the mitochondrion, for example, and ancestrally, if we go back a couple billion years, they would have had a complete suite of genes, cellular genes, for carrying out cellular metabolism. But over that 2 billion years, many of those genes have degraded, and they rely instead on nuclear genes. Mitochondria are no longer capable of living independently of the cell. So yeah, if you get a pipette and go in there and suck a mitochondrion out and try to culture it, no, it's not going to work, because it'll be missing a whole bunch of genes that it relies on the nuclear genome for. Now, what's illustrated over here, this is a map from the National Genographic Project, but you can also get similar things from 23andMe and all these other sorts of DNA sequencing programs. And what it's showing is, let's see, in orange, right there, this is a series of haplotypes, variants in mitochondrial DNA structure that allow us to track the spread of human beings across the planet. So we all arose in Africa down here, Eastern Africa, and you can find these variants. That's where all these little letters mean, M89, M201, HVVH, etc. Those represent mutations that arose in the mitochondrial genome and were passed on maternally to all the descendants of those individuals. And so this is a fun one where you can see orange is the maternal lineage. This actually happens to be from my sequence that I submitted. So I can see my many times great-grandmother was down here in East Africa and moved up here into Turkey and then up into northern Eurasia and then spread into Scandinavia. That's where my mom's family comes from. In blue is a different pattern. This is the Y chromosome inheritance pattern. So we'll get back to this later in the course, but Y chromosomes are kind of neat because they're only passed on from father to son, not to daughters. And so you get a patrilineal arrangement. So there's my many times great-great-great-great-great-grandfather back in here, and his family's migrating up through here, and they actually did this route. Again, we can trace that by looking at the variations in the Y chromosome, and we can trace them all up into here. They spent some time in Asia, and then ended up over here, this big pink circle. Yes, I am M343. That's the haplotype of my Y chromosome, which ended up in Spain and France and also England, which is my father's family came from. So you can see you can do fun things by tracking these. We'll talk more about that later in this course, I hope. Okay, mitochondrial DNA though. Here's a diagram of the mitochondrial DNA. It's short, it's a circular DNA. It's only 17,000 bases, to put that in perspective. Remember, the nuclear DNA is about 3.3 billion bases. So it's a little short thing. It's got ribosomal RNA genes on it. So remember, I just told you that they make their own ribosomes. They're different, distinct from the nuclear ribosomes. They have a set of 22 transfer RNAs. You all remember what transfer RNAs are, right? You didn't forget this from cell biology, I hope. Okay, so yeah, we got all these transfer RNAs. So we got all the machinery to do protein synthesis inside the mitochondria. And then there's a set of protein coding genes for oxidative phosphorylation. Again, you got to think back to cell biology. What was oxidative phosphorylation? Yeah, that was all the electron transport stuff. Remember that? So it's how we make energy. So mitochondria basically become specialized to just carry out these energy production mechanisms. So that's what we got there, a bunch of these. Now, I don't want to neglect chloroplasts. We have a lot of botanists in this discipline, you know. So here's chloroplast DNA. It's bigger. Look at that, 120 to 220 kilobases. It also has its own ribosomal RNAs. It's got its own transfer RNAs. It's got a bunch of protein coding genes for various functions. So chloroplasts have retained more of their ancestral abilities than mitochondria have. And 20 of these are specifically for photosynthesis and electron transport. And again, remembering back to cell biology, I know this is terrible. I'm asking you to remember things from maybe a whole 2 or 3 months ago, right? Oh man, it's been 4 months. Oh, you've probably all forgotten it, right? Anyway, remember in chloroplasts, we got all these these photosynthetic functions that are going on. And they also use electron transport chain to generate energy. So we've got all these fancy things going on inside these organelles. Can we see the effects of these? And yeah, we can. This is a great example. This is variegation this is called a 4 o'clock plant. Has anybody seen a 4 o'clock plant? I don't think they grow real well up here. But this is Mirabilis jalapa. It's got this interesting variegated pattern to its stem and leaves. So it's kind of white mottled with green, or green mottled with white, depending on where you are. And when you look at the plant carefully, what you can find is sometimes you'll find a branch that is all green. Sometimes you'll find a branch that is all white. Sometimes you'll find other branches that have a mix of green and white. And what's going on here? How does this happen? It turns out that this is caused by a defect in the chloroplasts. So, this is a complicated table, but I'll make it really simple for you. So what we've got here is a plant where parts of it are green, parts of it are white, parts of it are variegated. And you can take cuttings from this. And if you take a cutting from a green leaf or stem, just a pure green one, and you plant that and get it to grow, you will get a solid green 4 o'clock plant. If you take one that's solid white, you get a solid white plant. If you take one that's variegated, then you get a more complicated mottled pattern. What's going on is that not all the chloroplasts carry this mutation. Some of them do, and some of them don't. So what can happen is if you pass along, if you take a cell that's got both kinds of chloroplasts, the healthy chloroplasts and the mutant chloroplasts, it can produce cells that contain a mottled mixture of green and white. If you take a cell that's only got the healthy green chloroplasts, it can only produce healthy green chloroplasts. Now, this table looks kind of messy, but it's really simple once you see it. So this is the phenotype of a branch-bearing pollen, and this is the one carrying the egg. And the shortcut is just ignore this. This doesn't matter. So the pollen does not contribute. Stupid camera is getting really annoying. Anyway, so to repeat that, the pollen-bearing plants don't transmit chloroplasts to their progeny. So we can just ignore that entire column. On the other hand, if we look at the phenotype of the branch-bearing plant, so this is the one that's got the egg, and you see white, green, variegated, etc. Look at the progeny. It's all exactly identical to these. So whatever the cytoplasm of that source cell carries produces the same effect in the progeny. So this again is clear evidence that there's some kind of cytoplasmic transmission that's coming through the egg of the plant that isn't transmitted by the pollen-bearing plant. So again, you can just put a big X right through there. Simplifies everything. And once you do that, this becomes white, green, or variegated, and you get white, green, or variegated out of it. This is a similar example. This is a green alga. This is Chlamydomonas. And Chlamydomonas can be streptomycin resistant. So there are varieties of this alga where you hit them with streptomycin, they die. Other variants where you hit them with streptomycin, they're fine. They don't care. So here we got this one here. And then up here in this diagram, again, it's needlessly complicated because they're going through all the possibilities. So the dark cytoplasm, that means it's a plant with the streptomycin resistance in its cytoplasm. White means it does not carry streptomycin resistance, so it's susceptible. In addition, there's a little plus and a minus here. In Chlamydomonas, females are called plus and males are called minus. Okay. Pretty straightforward there. They are otherwise indistinguishable. You can't tell a male from a female. Okay. But they just have these properties. They also have another curious effect. So Chlamydomonas can go through meiosis, produce haploid spores, that then fuse to make a diploid organism. When they do that, it's equal contributions. Doesn't matter if you're male or female. You got a big fat gamete. Okay, am I going to have to do this every 3 minutes? Hope not. Anyway, you got a big fat gamete from the male and a big fat gamete from the female. They fuse. They're both contributing things to the daughter cell. But one of the properties of this plus is that those cells produce a collection of enzymes that destroy the cytoplasmic component of their partner. So they're both contributing equally, but the plus females are wiping out the cytoplasmic contribution from the other cell. And so that's all we see here. This is way too complicated. It's a really simple effect. So here we've got a bunch of cells that are strep resistant. Half are male and half are female and half are male. And we just do some crosses here. So we take this individual and cross it to a sensitive, streptomycin sensitive cell that is negative, and all of the progeny are going to have these particular arrangements. So we're going to have males and females, but they're all going to be strep resistant. Why? Because they got that from the female parent. Similarly over here, we do a similar cross, and in this case, the female parent is carrying the strep resistance, or the male parent is, but it gets wiped out by the plus here, so all the progeny are strep sensitive. You get the idea. So it's the same sort of thing as we saw with mitochondrial inheritance. Another example. Yes, today I'm talking about all these little tiny single-celled organisms, right? We haven't done much of that in this course, but we should. There's been a lot learned from the genetics of things like yeast and Chlamydomonas and Paramecia even, and all these things. In yeast, they discovered these things called Petite Mutations, and you can see the difference there. You put your yeast on a Petri plate with some nice nutritious agar, and the Petite mutants don't grow very well. They're not able to carry out certain steps of the metabolic pathways. So you get tiny little colonies compared to these normal colonies. And once again, this can be produced by the inheritance of cytoplasmic factors, in particular of mitochondria. Now, what we're seeing here, again, is another complicated diagram. And it's complicated because there's different ways this can get passed on to the progeny. Okay, this first column over here, these are segregational mutations. You take a haploid petite, a haploid normal, you cross those, you get a diploid zygote who is fine, who grows normally. Then it goes through meiosis to produce haploid ascospores. That's just what we call those yeast progeny. And what you find is Mendelian segregation. Half of them are petite and half are normal. That's what we'd expect for a new... I'm going to take this camera apart and figure out what's going on. Anyway, so this is the segregational mutants, are mutants that have affected the nuclear DNA. Then we get these two other categories, neutral and suppressive. Those are not mutations. Behave yourself. Yeah, we've only got, what, 8 people here? All those others in the class are going to depend on this, so let's not fail on them. Okay, anyway, so as I was saying, we got neutral and suppressive. Those are both mitochondrial mutations, and they have different effects. So neutral, when you do the same cross you did over here, you get a diploid zygote that's normal. And then when you look at the progeny from meiosis, they are all normal as well. So this is called neutral because the mutant doesn't have any kind of advantage. So the healthy, normal mitochondria take over and replace all the mitochondria in the daughter cells. So they're all healthy, they're all fine. Over here is a suppressive mutant. So same experiment, we're just crossing haploid petite with haploid normal. We get a zygote that's usually normal. And then when we look at the progeny, in this case, they're all mutant, they're all petite. And that's because the mitochondria take over, the mutant mitochondria. We don't call that dominant. This is a different phenomenon than dominance. But yeah, they're going to be overcome by all these defective mitochondria and not do as well. More. We can see this in fungi as well. So this is Pokie and Neurospora. And it's called Pokie because it makes them grow slowly. Okay. So if you've got the Pokie mutation, you're going to grow very slowly. It's maternally inherited. If you've got a female Pokie fungus and you cross it to a wild-type male, you get all Pokie. If you cross a male Pokie with a wild-type female, they're all wild-type. So there's that normal pattern of maternal inheritance. We can isolate mitochondria from these. And then we can take those mitochondria, suck them out of a defective Pokie fungus, and inject the mitochondria into a healthy cell and turn it Pokie. So that's pretty clear evidence that this is caused by mitochondria. And it's also that Pokie is suppressive. When you're doing that injection into a healthy fungus, it's already got mitochondria, normal healthy mitochondria. You inject the Pokie mitochondria in there and they screw everything up for everyone. So the Pokie spreads through that cell. So they disrupt normal mitochondria. And you're saying, okay, I'm tired of all these little single-celled organisms. Who cares about bacteria and fungi and plants? No, we don't encourage that attitude here. But yeah, we are kind of interested in human inheritance. Are there humans with mitochondrial diseases? And yes, there are. So this is just a general diagram illustrating a collection of effects that you typically see in individuals who are born with defective mutant mitochondria. They form a catalog of things that can go wrong. So here we see all these things that are really common in individuals that have mutant mitochondria. We see, first of all, let me just mention that some of these things are more common than others. So brain, eye, ear, and heart are really active organs, high metabolic rates. As you might guess, they rely a lot on mitochondria to carry out their functions. So a lot of the defects you see in individuals with these mitochondrial diseases affect those organs. So you get encephalopathy, you get deafness, you get blindness, you get cardiomyopathy, all kinds of problems here. But it can also affect other cells, like, for instance, hormonal cells, glands. The kidney is also a very metabolically active region. So is the liver. We have muscle weakness. All kinds of things can occur if you've got a mitochondrial disease. It's not a good thing to come down with. You may have heard in the news that there are various experiments that have been carried out where they try transplanting mitochondria from a donor to an affected individual, usually during implantation of a new embryo. You try to give it an edge by giving it cytoplasm from an individual who does not have one of these mitochondrial diseases. So there's been some of that going on to prevent this stuff from happening. Because as you can see, there's a lot that can go wrong here. All right, there's a whole bunch of these. And if you read the medical literature on mitochondrial diseases, you will be overwhelmed with acronymitis. It's like they've all got different acronyms. So here's a common type. This is called myoclonic epilepsy and ragged red fiber disease, or MRF. It's one of the more common mitochondrial diseases. If you look at how this gets transmitted, the offspring of affected mothers are also affected. And I'll come back to that in a moment, because notice, you've got women who have this disease and who survive to adulthood and have children. How do they do that? Because it sounds like losing your mitochondria is pretty disastrous. You also find that offspring of affected fathers are perfectly normal. These are all hints that it's being passed on maternally. The effects of MRF are dementia, deafness, and seizures. Also implied here, ragged red fiber refers to the histology of the muscle tissue. So you've got ragged red fiber, there's breakdown of some of the fibers in the muscles. And the cause of this is a mutation of one of the mitochondrial tRNAs. Yeah, we mentioned this in cell biology too. What would happen if you mutated a tRNA so that, for instance, instead of at the appropriate codon, you don't put glycine, you put valine or something? That's disastrous because it leads to changes all across the genome, the expression of the genome. And this is the case of that, where they've got a damaged mitochondrial tRNA. It messes up the translation of all those mitochondrial genes. And so they're going to have problems. Again, remember, mitochondria are really important for energy transduction. So if we want to extract energy, we need those mitochondria. And having this kind of global damage to the mitochondrion is not good. Another acronym, Labor's Hereditary Optic Neuropathy, LHON. Again, just like the previous one. This is one of the giveaways, is you look at the pattern of inheritance. If it's inherited only from mothers and not from fathers, that sort of suggests that there's a maternal effect translation going on here. In this case, the effect is late onset blindness. So kid is born, looks fine, goes to school, grows up, and then later in life, eyes shut down, they stop working. So there's gradual accumulation of defects there. In this case, the cause is mutation of one of the mitochondrial oxidative phosphorylation genes. So they've lost one of those electron transport pathway proteins. Okay, so that's LHON. We got some more acronyms coming up. They're all going to start sounding the same. This one is called Kernstar Syndrome. First two. Yeah, this is happening over and over again. You should recognize this. When, yes, offspring of affected mothers are affected, offspring of affected fathers are normal. In this case, what you have is progressive loss of hearing and vision and heart disease. I told you heart's kind of a sensitive organ to this. Okay, so and hearing, vision, these tissues are highly metabolically active, and so they're going to be more sensitive to these kinds of problems. And the cause here is multiple deletions in the mitochondrial DNA. So we've got multiple breakpoints in the mitochondrial DNA, so they don't properly synthesize the necessary proteins. Yes, I have some more. Oh yeah. If you're a pre-med, you might have to learn these someday. Okay, but in general, for this class, we just need, it's a welter of acronyms. They all kind of make sense. So we've got NART, MALIS, and MMC. These all have myopathy, so muscle weakness, hearing loss, epilepsy. Yeah, epilepsy is another common effect of things that affect the brain. Ataxia, that means troubles in coordination, things like walking, so forth. They have tremors, spasticity, eye abnormalities, and abnormal lactate and or pyruvate levels. which tells you that one of the ways we can test for these is by looking at blood metabolic products. So as you might expect, if the mitochondria are misfiring, or they're malfunctioning, you're going to get changes in things like those essential molecules, lactate and pyruvate, that we talked about so much in cell biology. Okay, here's the acronym meaning. No, don't memorize these. Unless you're pre-med, then you got to memorize everything, but yeah. Okay, neuropathy, ataxia, retinitis pigmentosa. Malus is mitochondrial myopathy, encephalopathy, lactic acidosis, and stroke-like episodes, and MMC is maternally inherited myopathy and cardiomyopathy. Yeah, so we find all these different syndromes, and like I said, doctors go acronym crazy, and they give them different little labels and get their name in a medical journal somewhere. They all seem to have subtly different causes, though. So different kinds of mutations in mitochondria. Okay, why aren't they all dead? Yeah, think about how much you rely on your mitochondria to generate energy. We talked about that in cell biology, right? Yeah, what happens if you just lose the oxidative phosphorylation pathway? Boom, you're going to be so inefficient, you're not going to survive. And this is generally true of most embryos who have a severe case of this. They don't make it to term. Okay, they don't even have the energy to divide properly. So you're not going to see them. When you see these in children and adults, it's because they're heteroplasmic. Okay, what does heteroplasmic mean? It means you have a mixture of different cytoplasmic properties. You've got multiple mitochondria in every cell, right? And most of us who are healthy have a mix of healthy mitochondria in there. But as you can see here, we've got different possibilities. If you've got a mutant mitochondrion, can you see that from back there? Yeah, so the red ones are supposedly mutant. They've got problems, they've got defects. And the green ones are good, healthy mitochondria. So you've inherited a defective mitochondrion. What will then happen is that through division, these mitochondria will multiply, and it's kind of chance what will happen. You could inherit a cell that's got a lot of defective mitochondria, and that one's going to be really severely affected. You could inherit a moderate amount, and you'll be moderately affected. Or you could be mostly free of the defective mitochondrion, and then you're in pretty good shape. You look unaffected. So these are all possibilities that can happen. And you'll have a range of these possibilities within an individual. So somebody who's got a defective mitochondrion that they inherited as an embryo, you can imagine you'll have very different effects if those cells end up mostly in the brain. Then you'll have all kinds of serious effects. On the other hand, if they end up in the muscles of the body, you're not going to be playing baseball very well. So lots of subtle variations there. It means that this is a kind of disease that has a huge range of degrees of expression, and is very problematic in that way. It also means that somebody with these particular problems, who's healthy enough to make it to adulthood, can have children, and maybe those children will be mostly unaffected. Or maybe you've got somebody who's only mildly affected, they make it to adulthood, and one of their children is severely affected. Just random chance. It's variable what you'll get out of this particular kind of trait. And that's all due to heteroplasmy. So all of us here might very well be heteroplasmic. We might have two different variant mitochondria in our cells. Hopefully healthy mitochondria, so we don't see the kinds of phenotypes we described earlier. Okay, here's another kind of heredity. This is called infectious heredity. And this involves transmission of a symbiotic or parasitic microorganism infesting the mother to the cytoplasm of her progeny. There's a scary one, isn't it? Yeah, you don't want to pass diseases along to your kid. We've got several documented cases of this in various organisms. So here's a common one. CO2 sensitivity in Drosophila. In Drosophila, hey, how have we been knocking out Drosophila in the lab? Yeah, y'all remember what we used to?
FlyNap.
Yeah, we use FlyNap. We use a chemical agent to knock them out. It's kind of annoying because it's stinky. A lot of people don't like that. It's a little tricky to use. It takes a few minutes to knock them out. In the big fly labs, no, they don't do that. They don't use FlyNap. Instead, what they've got is they've got CO2 lines running through the lab. And so you get a nice little hose with a little spigot that you can control, and you just take your bottle of flies and you spritz in some carbon dioxide, and they pass out. They suffocate and flop down. This is actually how I knock out spiders, for instance. I don't have a CO2 line. Instead, what I've got is a baking soda volcano. You know, you take vinegar, you mix it with baking soda, and it produces CO2. And a little hose coming off that, and I run that hose into my spiders, and they pass out for a few minutes so I can do my wicked experiments on them. Anyway, so CO2 is a really common and routine way to knock out invertebrates. And it doesn't do them any harm, as near as we can tell. They pass out, they're suffocated basically, and then a little later they wake up. And the transitions are pretty quick. So that's the big advantage there. So in all these big fly labs, they got these CO2 lines, and they start gassing their flies to knock them out. Normal flies, when you hit them with a little CO2, they pass out, they stop moving, you do your experiments, you're done. Trust me, our experiments would be easier if we could have CO2 lines in the lab. You just spritz it in there instantly. All the flies are knocked out and you can do your experiments. Okay, so normal flies do that. If you expose to high concentrations, you get temporary unconsciousness, again, what you want. There are sensitive flies, though. If you hit them with a low dose of CO2, they go into spasms, they start twitching. You've seen flies twitching, right, when you knock them out. This is worse. They're doing a little dance down there. And if you say, okay, well, I've got to get these guys asleep, and you jack up the concentration a little bit more, they die. That's not what you want. That makes the experiment kind of difficult. It turns out that this sensitivity is transmitted by the maternal parent by the way of a virus called sigma. So if mom has sigma, it gets passed on. Yes?
Oh, sorry, I was just stretching.
Just stretching, that's okay. Okay. So the virus is the virus is conferring the sensitivity to CO2 to these flies. Okay, again, passed on maternally. Now here's another really wicked one. Oh, this is just pure evil, right? There is an infectious agent that causes all the males to die. Oh man, yeah. Have you seen that? There's that series Y, The Last Man. Has anybody seen that? It's also a comic series. Oh, it's a fictional story about all the men dying off and the world being run by women. Yes, I can tell you're getting into that. That's a great idea. But anyway, there are things that do that. So this is an infectious agent that will produce a toxin that's lethal only to the males. So all the male flies die off, the female flies are fine. And in this case, it's called by a protozoan. There are various single-celled organisms that can actually populate the cytoplasm of large cells like egg cells and thrive in there. And these, they're killing off all the guys. So if you're fantasizing about that, there's ways it could happen. Okay, here's another one. This is called kappa. Paramecia, I have not talked about their genetics. I don't know if I want to because it is really messy. So here's Paramecia, and you've all seen Paramecia swim around, those slipper-shaped things with the cilia that swim around. They carry multiple nuclei. So they carry a macronucleus, and they carry a pair of micronuclei. And those are all diploid. So single cells with 3 nuclei. And one of the nuclei is very different from the others. If they are going to carry out replication, if they're going to reproduce, they can do this by swapping micronuclei with a partner. So they'll actually conjugate like this, they'll line up, and the micronuclei will go through replication. We go from 2 to 8. Oh, that sounds like meiosis, doesn't it? So they go through this meiotic process. They produce 8 of the micronuclei. The macronuclei just sits there looking on. And then what will happen is they will disintegrate most of the nuclei. And then they'll pass on a pair to their partner. And then that partner's nuclei will start to expand and grow and replicate, and you get back to the final state. Yeah, it's complicated. So I promise I won't introduce that into this course. Take a bacterial genetics or microbial genetics, and they will go on at length about these because they are so bizarre and so interesting and so very different from us. Okay, so we got these Paramecia that can carry out sexual reproduction. That's what we're seeing here. This is what sex means to a Paramecium. But they can also replicate. They can also just divide all by themselves. They don't need a partner. So we got 2 modes of replication going on in these organisms. Come on. You can do it. Yes. Oh, too fast. Too far. Go back. Okay. So here's a complicated phenomenon. So we're going to go through these conjugations. We're going to swap genetic material between 2 Paramecia. In addition to swapping nuclear material, though, they can also sometimes exchange cytoplasm, but not always. So here on the left, this is a conjugation that doesn't involve swapping any cytoplasm. They just swap the nuclear material. Over here is a conjugation. This one is a sloppy conjugation because you swap the nuclear material, but you also pass along little bits of the cytoplasm. So we're swapping fluids as well as nuclei. Okay, so we got these 2 kinds of conjugation. In addition, what we're seeing here is that there is something called kappa. Kappa is a particle that's found in some of these Paramecia. And kappa does 2 nice things for the cell. One, it gives it immunity to kappa. And 2, it secretes the kappa toxin, which kills all the other Paramecia in the neighborhood. Okay, so that's why it's called killer. If you put one of these Paramecia in a test tube with other non-kappa Paramecia, it starts secreting this stuff, and all the other Paramecia die off. And then the killer can feast on their remains, I guess, or whatever grows from their remains. So here's a case where you've got this exchange. We're also, in this case, swapping a particular nuclear agent, this K. So big K is necessary for the kappa particle to survive. So we swap in all these particles. We also exchange the big K. So what you get is progeny that in this case, this one still has big K, so it can support a population of cytoplasmic killer particles. This one has big K, but it didn't exchange any cytoplasm, so it doesn't have them. Over here is a case where we've got cytoplasmic exchange, and same situation, same genetics. So we get progeny that are both big K, little k, and in this case, both have the kappa particle. And then what we can do is take those paramecia and have them reproduce asexually by autogamy. And what do we get? Well, this one's only going to produce sensitive ones because it doesn't have the kappa particle. But half of its progeny could, in principle, support a kappa particle. And over here we see autogamy that produces progeny that half will have big K, and kappa particle. This one is big K, or little k, so it's only got the recessive K. It will have inherited some of the kappa particles from its parent, but they'll die off. Without big K, the kappa particle dies. So there's a kind of synergy between nuclear genes and these cytoplasmic factors. So it's a fun game to play with, thinking about all the steps that are going through here and how this can spread through a population. Okay, we got 10 minutes. I can get started on this. Maternal effect. Maternal effect is where the genotype of the female parent determines the phenotype of the progeny, regardless of the genotype of the progeny. Yeah, so in this case, mom is making something that is necessary for the normal development of the progeny, but it's passed on through the cytoplasm, not through the nuclear genes. Here's an example. So this is a festia. It's a little maggoty little beetle larva. And we've got two variants here. We've got an albino form that's got a red eye, okay? And this is the wild type form. It's got a brown eye. And in this case, this is a heterozygote. This is a homozygote here. And we're going to cross these. And the sex of the parents matters. So in this case, we're going to have a female that is little a, that's this one, and a male that is heterozygous and brown. We're going to cross those and we'll get some larvae, and we look at the color of the eyes, and what we find is that some of the progeny, as you might expect from this cross, they're big A, little a, right? Half will be big A, little a, and half will be little a, little a. So the half that are big A, little a contain males and females, and they all have brown eyes. The progeny that are little a, that's all of these over here, contains males and females, and they all have red eyes. Okay, so far so good? So we're transmitting some kind of albinism factor here. Over here, what we do is we change the sexes around. So now we've got a female that's brown-eyed and a male that's red-eyed. It's the same cross, we've just changed the sex. Now when you look at the larvae, what you find is you get some that are big A, little a, male and female, and they're brown-eyed. And you're saying, okay, that's to be expected. That's what I would expect to see, because you got a big A there, right? Then we get some that are little a, over here, males and females, and they're all brown-eyed. How do you explain that? Little a, it's supposed to be red-eyed. This is little a, and it's brown-eyed. what's going on? It worked over here, but it didn't work over here. Anyone have any ideas to explain this? I know, it's a new complication on sex-linked inheritance, isn't it? Sometimes it works and sometimes it doesn't. Well, to spill the beans and tell you the answer, what we think is going on is that the female, whatever her genotype, is producing a package of RNA and proteins that get into the eggs. Makes sense, right? You got a female, you make these big, fat, juicy eggs that are just full of all kinds of goodies for the embryo to grow on. And if your female is brown-eyed, she can load up that egg with all the brown pigments, brown pigment RNA, whatever, so that when the egg hatches out, it's got brown eyes. In this case, we've got a female who cannot make that RNA. So she cannot synthesize the brown pigment RNA, can't pack it into the egg. The male, the worthless, useless male, is just producing sperm that have almost nothing on them, so they don't have it. They don't provide the pigment. So in that case, you'll fail if you can't synthesize it zygotically. I like to think of this as, okay, mom has the job of packing a lunch for her babies, right? So she's going to pack a lunch with whatever she's got at hand. She's going to pack this nutritious lunch. So she's going to make a nice bologna sandwich for her babies, unless she doesn't have any bologna, right? And then what she's going to give them is just bread. Ugh. So that's all these individuals get is just the bread, no bologna on it. In this case, the female can't pass on the gene to allow them to make their own bologna, but she can make the bologna herself to feed the kids. Okay, so this is an interesting phenomenon. Then we got to ask, okay, what about these when they grow up? So this individual, let's just look at the females. We got a little a female. She's going to grow up with brown eyes, not red, and then she's going to have babies. What will she pass? What color will the eyes of her babies be? So she gets to adulthood. She's sitting there making eggs. She's filling those eggs with nutritious goodies. But her genotype is little a, right? She can't make any bologna. So the kid doesn't get any. So she will have progeny who are red-eyed no matter what color eyes her mates have. Whoa. Yeah, maternal effect is fun because basically what it does is it displaces the phenotype from the individual that has it, who is you're looking at, who has the genotype, to their progeny. So it's like everything is delayed one generation when you look at the phenotypes. Here's another famous example. These are snails. Have you ever looked at a snail really closely? Some snails are right-handed. That is, that shell is coiling that way. It's got a right-handed coil to it right there. And some shells are left-handed. See how it coils this way with my left hand? So we've got these differences. This turns out to be very important to snails because when snails mate, they kind of clamber on each other and they try to align their openings. And if you're a right-handed snail trying to mate with a left-handed female, no, it doesn't work. Nothing lines up. So there's actually selection going on for a particular handedness. But anyway, we can find these snails. We find dextral and sinistral snails. And we cross these. Give them time. They'll figure out how to line everything up. So we're going to cross these two. And then we ask, okay, what do the progeny look like? All right, so in this case, if we cross a dextral, right-handed egg, a female, with a left-handed male, all of the progeny are right-handed, okay? So we get right-handed snail. It doesn't matter what the male's contribution is. Over here we see we cross a right-handed sperm with a left-handed egg, and we get all sinistral, all left-handed. Making sense so far? And that's despite the fact that these two are genotypically identical. They're both heterozygous. They're both big D, little d. But their shell coils in a different way. And then the fun begins. Because with snails, we can also do self-fertilization. We can ask this individual to mate with itself and produce progeny. And we ask, what kind of progeny will it produce? Well, genetically, you'll get a Mendelian mix. Look at that, big D, big D, big D, little d, big D, little d, little d, little d. You'll get that nice 3-to-1 ratio, except this individual is little d, and it's got a right-handed coil. Why is that? It's because it inherits the coil of its parent, of its maternal parent. Doesn't matter what its genes are. In this case, mom packed in a left-handed wrench or a right-handed wrench. So you develop that particular way. Okay, but in this case, come back. But in this case, all right, it's little d, which ought to be like that. But in early development, it inherited the factors to make a right-handed coil. So it looks like this. And when it has babies, all of its babies are left-handed. Likewise, we can look at this other pattern. It's exactly the same. What matters is the genotype of the mother determines the phenotype of the offspring. Kind of mind-blowing if you think about it. So, and I'll briefly explain that before I let you go. The factors that determine shell coiling in snails are expressed really, really early in development. So here's a 2-cell snail egg, and then a 4-cell. And at the 8-cell stage, it divides to produce 4 cells, and then those 4 cells rotate just a little bit. And they rotate in a consistent direction. So this is where the coiling comes from. It's from this one little rotation. And in some snails, it rotates to the right, as you see here. In other snails, it's going to rotate the other way, to the left. And that has profound effects on later development and determines the coiling of the shell. Okay, that's a good place to stop. I will review this again on Wednesday and make sure it sinks in. And we haven't even gotten to the hard part yet. We haven't talked about imprinting. Yay.